So I've spent nearly 25 years working on the Human Genome Project. I started my career at the world-famous Wellcome Sanger Institute in Cambridge, United Kingdom, and we were responsible for generating around 1/3 of the human genome, and this is where I completed my PhD. I also led the gene analysis for human chromosome 10, which was published in Nature in 2004, and was part of the ensemble team that create transcript models that are used in research and clinical diagnostics. However, after 23 years at Sanger, I made the move to Congenica, where I'm currently the clinical domain lead for epilepsy. And I've been at Congenica for just over 2 years. I also lead an international collaboration that's investigating the genetics of early infantile epileptic encephalopathies. A bit of a mouthful, but they are a group of severe and potentially fatal epilepsies that occur in very young children. So a little bit about Congenica. We are based on the Wellcome Genome Campus near Cambridge, next to the Wellcome Sanger Institute. And with world-renowned scientific and clinical expertise, we specialise in rare inherited and de novo genetic diseases. We are also a clinical interpretation partner for the UK 100,000 Genomes Project, which is a national project to sequence 100,000 genomes for people with cancer and rare disease. So what makes us unique is that we have combined great bioinformatics and scientific experience with expert clinical experience. Our founders are bioinformaticians and scientists from the Wellcome Sanger Institute who are experts in understanding large volumes of complex biological data, as well as clinicians from Great Ormond Street Hospital who are experts in the field of genomic medicine. So they therefore understand the needs of doctors trying to provide patients with a diagnosis. And since the founding, Congenica has built a team around them which includes software developers, data management experts, and a clinical team. So now a few facts about epilepsy. Epilepsy is characterized by recurrent seizures due to abnormal synchronized neuronal firing in the brain. But epilepsy is a neurological condition, which means it affects the brain and the nervous system. However, it is also a physical condition because the body is affected when someone has a seizure. There are over 40 different types of seizure, and not all seizures involve convulsions, unconsciousness and shaking. So for example, around 3% of people with epilepsy have photosensitive epilepsy. And here I have included on this slide a warning sign from the recent film Incredibles 2 that warns people with photosensitive epilepsy that they may be triggered from the events in the film. There are more than 30 types of syndrome, which include some of the most severe and debilitating forms of epilepsy that are seen in very young children in the first months of life. And they include Ohtahara syndrome, which is what ex-British Prime Minister David Cameron's son Ivan tragically succumbed to, Dravet syndrome, and West syndrome. So over 500,000 people in the United Kingdom have epilepsy, which is quite a lot. It's about 1 in 100 or 1% of the population, which makes it a common disease, although individual causes of epilepsy may be very rare. So more than 3 million people in the USA and more than 60 million people worldwide have epilepsy. 1 in 20 people will have a one-off epileptic seizure at some point in their life, although this doesn't necessarily mean that they have epilepsy. 1 in 50 people will have epilepsy at some time in their life, although not everyone with epilepsy will have it for life. Around 87 people are diagnosed with epilepsy every day in the United Kingdom. And epilepsy can occur at any time of life and to anyone of any race, sex or age, although it is most commonly diagnosed in children and in people over 65. So it is very unpredictable. There are also problems with misdiagnosis, which occurs in between 20 to 30% of patients. And in the UK, there are around 1,000 deaths per year as a result of epilepsy, of which 42% are potentially preventable. It also places a great strain on the National Health Service, costing in excess of £1.5 billion a year. And epilepsy is second only to stroke as the most common neurological disorder. So for comparison, the total cost of epilepsy in the United States is estimated to be around $15.5 billion a year. The psychosocial consequences of epilepsy and the permanent state of being epileptic are often more problematic than the seizures themselves. So issues that commonly arise for patients and families include death, problems with friends and relationships, family planning, parenting, accidental injury, and schooling, partly due to problems that people with epilepsy may have with learning. So the lack of predictability of seizures is a major cause of loss of self-confidence and self-esteem and undermines the confidence of people with epilepsy. Patients with epilepsy are more likely to have vascular and autoimmune problems and cancers, and this is possibly due to lifestyle choices such as smoking, being overweight, or high cholesterol. Also, for the parents, there is a tendency for overprotection or overdependency as a result of guilt and fear of their child being more susceptible to danger. So the causes of epilepsy can include a structural change in the brain, such as perhaps the brain not developing properly, or damage caused by a brain injury, such as a car accident, infections like meningitis, or a tumour. And causes can also include structural changes due to genetic conditions, such as tuberous sclerosis or neurofibromatosis. And of course, epilepsy can be caused by many other genetic factors. So if we go back to the year 1975, most epilepsies were characterized as unknown or idiopathic. Now the term idiopathic is no longer used. In many ways, it was a bit of a fudge to cover up the fact that no one really knew what the causes were, and the causes were providing an incorrect reassurance to patients that a diagnosis had been made. However, technological advances, such as MRIs, have allowed us to view the brain at a much greater resolution, identifying lesions that were not previously identifiable. And even more importantly is the impact of genome sequencing that has revealed that a large proportion of previously unknown epilepsies now have known genetic factors, including single-gene epilepsies, epilepsies with complex inheritance, and modifiers and susceptibility alleles.